Hello viewers, welcome back to our channel. I am Dr. Rupamani Thakur, and in today's presentation I will be discussing about a very interesting topic of cell biology, that is the cell cycle and cell cycle checkpoints. The presentation has been designed to address different aspects of the cell cycle which will help us to understand the necessity of the cell cycle, summarize the different phases of the cell cycle, and understand the significance of cell cycle checkpoints which play an important role in cell cycle regulation. With these objectives in mind, let us begin today's discussion on the cell cycle and cell cycle checkpoints. It is well known that self-reproduction is one of the fundamental characteristics of cells. Cells divide in order to grow, repair tissues, and reproduce themselves. This is achieved by duplicating their contents and then dividing in two. The term cell cycle refers to the ordered series of events that ultimately lead to cell division and the production of two daughter cells, each containing chromosomes identical to those of the parent cell. In other words, the cell cycle can be simply defined as the sequence of events required for a single round of cell division. The cell division cycle is the fundamental means by which living things are propagated. In unicellular organisms such as bacteria, each cell division produces an additional organism. But in multicellular organisms, repeated cycles of cell growth and division are required. Though the details such as the duration of the cell cycle varies in different types of cells, but certain well-coordinated requirements are universal. Essentially, the cell cycle of most cells consists of four coordinated processes. These are cell growth, DNA replication, distribution of the duplicated chromosomes to daughter cells, and finally cell division. With these brief idea about the cell cycle and its necessity, let us now discuss about the different phases of the cell cycle. The concept of cell cycle and its different phases were first ascribed by Alma Howard, a radiobiologist, in collaboration with Stephen Pelk, a physicist, in the year 1953. According to Howard and Pelk, cells that are dividing undergo a series of coordinated events called the cell cycle and is traditionally divided into four distinct phases. These are the G1 phase, the S phase, the G2 phase, and the M phase. As observed under the microscope, the cell cycle is essentially divided into two basic parts. The non-dividing part, which is called the interphase, constitutes about 95% of the cell cycle and includes the events of the G1 phase, S phase, and G2 phase. And the dividing part includes the events of the M phase. The key molecular events taking place during the cell cycle are DNA synthesis and chromosome segregation. DNA synthesis occurs during the S phase of the cell cycle, and chromosome segregation, which is a result of mitosis or nuclear division followed by cytokinesis, occurs during the M phase of the cell cycle. Both these phases, that is the M phase and the S phase, are separated by two gap phases called the G1 and the G2 phase. The G1 phase corresponds to the interval between the end of mitosis and initiation of DNA synthesis. The G2 phase is the interval or gap between the end of DNA synthesis and the beginning of mitosis. Both the gap phases provide additional time for cell growth and checks whether the necessary materials and energy is available to proceed to the next phase. A typical mammalian cell divides approximately every 24 hours, where the G1 phase lasts for about 11 hours, the S phase lasts for about 8 hours, the G2 phase lasts for about 4 hours, and the M phase is the shortest and it lasts for only about 1 hour. But there are other types of cells which divide much more rapidly. For example, it takes only about 90 minutes in budding yeast to progress through the 4 stages of the cell cycle. Some cells cease proliferation and enter a quiescent stage called the G0 phase, and the cells in this phase are known as the resting cells. We will be discussing about these phases in more details subsequently in this presentation. But before I move on, I would like to mention that the cell cycle that we are discussing is also known as the mitotic cell cycle. This is because most eukaryotic cells, such as somatic cells, divide and maintain the number of chromosomes in the daughter cells that were present in the parental cell. Let us now discuss about the phases of the cell cycle in more details. The G1 phase or the gap 1 phase constitutes about 30 to 50% time of the cell cycle or lacks entirely in rapidly dividing cells. For example, in early embryonic cell cycles, the egg cytoplasm rapidly divides into smaller cells. These cells do not grow during these cycles, and there is no G1 phase or G2 phase, and it only consists of short S phases alternating with M phases. It is important to note that during the G1 phase, no DNA synthesis occurs. Instead, this phase involves the synthesis of different types of RNA, that is, messenger RNA, ribosomal RNA, and transfer RNA. This phase also involves the synthesis of different types of proteins and membranes which lead to the growth of nucleus and cytoplasm of each daughter cell towards their mature size. So what are the proteins that are synthesized during the G1 phase? These are the regulatory proteins which control various events of mitosis. Now these regulatory proteins are very important and they play a very important role in regulating the cell cycle. We will be discussing about these proteins in a separate presentation. The G1 phase also synthesizes the enzymes that are necessary for DNA synthesis. One very important enzyme is the DNA polymerase. And it also synthesizes the mitotic apparatus proteins such as tubulin. The S phase or the synthesis phase follows the G1 phase and occupies 35 to 45% of the cell cycle. Two key events occur during the S phase of the cell cycle. The first one is replication of DNA and synthesis of histone proteins. During replication, each DNA molecule is duplicated. Each resulting DNA molecule is called a chromatid, and the pair of duplicated DNA molecules is called sister chromatids. The second important part of the S phase is sister chromatid cohesion. The sister chromatids are held together by the process of cohesion with the help of the protein complex called cohesin. This state of cohesion is very important as it links the sister chromatids together, and the cohesion ring is destroyed only during chromosome segregation, which occurs during the M phase of the cell cycle. So in summary, at the end of the S phase, each chromosome has 2 DNA molecules and a duplicate set of genes. The G2 phase or gap 2 phase follows the S phase of the cell cycle and occupies 10 to 20% time of the cell cycle. The G2 phase is a period of rapid cell growth and protein synthesis. That is, this phase prepares the cell to undergo mitosis and cell division. Another important aspect of this phase is that microtubules, which play a very important role in cell division, reorganize to form spindles. The G2 phase is followed by the M phase or the mitotic phase. Mitosis starts at the culmination point of the non-dividing phase of the cell cycle, that is the interphase. Cells that enter mitosis are required to have successfully copied their DNA and rest of the content of the cell. The mitosis is divided into several phases. These are prophase, metaphase, anaphase and telophase, followed by cytokinesis. The different phases of mitosis and the significance of this particular cell division will be discussed in a separate presentation. So basically, during mitosis, the sister chromatid cohesion is removed by destruction of the cohesin ring, which was formed during the S phase of the cell cycle. This allows the sister chromatids to get segregated to opposite poles of the mitotic spindle. Another important aspect to remember about the M phase is that the chromosomes during this phase are apparently too condensed to allow any form of transcription. It is important to note that many cells cease proliferation and enter a quiescent stage called G0 phase. In multicellular organisms, most differentiated cells, such as the nerve cells and cells of the eye lens, exit the cell cycle and survive without dividing again in the entire lifetime of the organism. Such post-mitotic cells generally exit the cell cycle in G1 phase and enter a phase called the G0 phase. However, there are some G0 cells that can return to the cell cycle. For example, cells of the internal organs such as liver, and they resume replicating. However, this re-entry is regulated, thereby providing control of cell proliferation. So far, we have understood that the different phases of the cell cycle have distinct activities associated with it. The key molecular events, that is DNA replication and chromosome segregation, in addition to cell growth, must be carefully regulated and coordinated in the cell cycle in order to ensure formation of progeny cells without any defect. Defects in cell cycle regulation are a common cause of abnormal proliferation of cancer cells, which is a major health issue in developed and developing countries like India. In short, Proper control of cell division is vital to all organisms. But how is this achieved? In the next few slides, we will be discussing about how the cell cycle is regulated. The progression of cells through the cell cycle is regulated by extracellular signals from the environment as well as by internal signals or control points that regulate progression through various phases of the cell cycle. Cells commit to a new cell division at a specific point in the G1 phase of the cell cycle, known as start in budding yeast or the restriction point in animal cells. If we consider the animal cell cycle, it is found to be regulated by extracellular signals such as availability of growth factors, which act in G1 phase at the restriction point. In the absence of growth factors, cells enter G0 or the quiescent stage. In case of budding yeast or Saccharomyces cerevisiae, the cell cycle is regulated by external signals such as nutrient availability, which act in G1 at the point which is known as start. As we can see in the image here, which shows the regulation of cell cycle of budding yeast, external factors such as nutrient availability, mating factors and cell size regulate primarily at a point in the G1 called the start. In case of animal cells, the availability of growth factors control the cell cycle at a point in G1 called the restriction point. If growth factors are not available during the G1 phase, the cells enter the G0 phase or the quiescent stage. In addition to the extracellular signals that regulate the cell cycle progression and commitment of a cell to undergo a cell division cycle, the different stages of the cell cycle are highly coordinated with one another. This synchronization between different phases of the cell cycle is reliant on cell cycle checkpoints, which prevents entry into the next phase of the cell cycle until the events of the preceding phase is accomplished. For example, the DNA has to be completely replicated before the cell undergoes chromosome segregation or mitosis. In short, the cell cycle checkpoint monitors and regulates the progression of the cell cycle. There are 4 cell cycle checkpoints. These are the G1S checkpoint, the S checkpoint, the G2M checkpoint and the M phase checkpoint. We will be discussing about the 4 cell cycle checkpoints in more details in the next slide. Let us now try to understand the significance of the cell cycle checkpoints, which acts as the internal surveillance mechanism. The first checkpoint, that is the G1S checkpoint, is a DNA damage checkpoint. The G1S transition is a rate limiting step in the cell cycle, and arrest at the G1S checkpoint allows repair of any damaged DNA before the cell enters the S phase or the synthesis phase of the cell cycle for replication. The second checkpoint is the S checkpoint, which is another DNA damage checkpoint. So this particular checkpoint monitors the integrity of DNA to ensure that damaged DNA is repaired before it is replicated. Arrest at the S checkpoint promotes the repair of any error that might have occurred during DNA replication, such as incorporation of incorrect bases or incorrect replication of DNA segments. The next checkpoint in the cell cycle is the G2M checkpoint, which is another DNA damage checkpoint. So this checkpoint prevents the initiation of mitosis if the cell contains DNA that has not been completely replicated or contains unrepaired defects. This particular checkpoint ascertains that the cell has adequate cytoplasm and phospholipids so that the cell can undergo cell division to form two daughter cells. The next checkpoint in the cell cycle is the M phase checkpoint. This is a spindle assembly checkpoint, and this maintains the integrity of the genome towards the end of mitosis. This particular checkpoint also monitors the proper alignment of the chromosome on the mitotic spindle, thus ensuring that a complete set of chromosomes is distributed accurately to the daughter cells after a completion of cell division. So far, we have understood how the external signals regulate the cell cycle and how the checkpoints monitor and regulate the progression of the cell cycle. But what exactly modulates the cell cycle checkpoints? The cell cycle checkpoints consists a group of regulatory proteins which are the master regulators of the cell division in all eukaryotes. These regulatory proteins supervise and control the progression of the cell through different phases of the cell cycle. I will be discussing about these cell cycle regulatory proteins in my next presentation. These are the books that has been consulted while preparation of this particular presentation and may be referred for further understanding of this particular topic. So this brings us to the end of the presentation. Drop in your queries and feedback in the email address given here. Thank you.